Okay, so experiment 11 was the gram stain, and it's a very important experiment because it'll be on your practical, and the written part. So they do the gram stain because it tests your aseptic technique, it tests your dyeing abilities, your ability to make a slide, and of course your gram stain stuff. And so it's a very general thing that has a lot of practices in it, and then it also is very important to microbiology because gram stains are, gram negative and gram positive bacteria are very important to distinguish between. So the next couple labs are going to deal with differential staining. So a gram stain differentiates between gram negative and gram positive bacteria. Endospore stain can tell you what is an endospore and what's not an endospore. Acid-fast staining, we'll get into that, but it allows you to differentiate between acid-fast cells and then just regular non-acid-fast cells. And so that's what the next couple labs are going to be covering, is this differential staining concept. So we're going to look at the gram stain in experiment 11, of course. So the general overall picture is that you put stain on all cells, and then you remove from the stain from one type of cell, but not the other type, and then you stain the rest of the remaining cells to where you have one cell is one color, and then another cell is another color. So if we look at our overall picture, if you go to structure and function of the study guides I sent you, you'll see some stuff about gram positive and gram negative cells. So there's a lot of, the way I remember is there's a lot of P's in this whole thing. So you have positive, purple is positive, and you have more peptidoglycan, and which is important. So all bacteria cells have a cell wall made of peptidoglycan, which is repeating units of N-acetylglucosamine and N-acetylmuramic acid, which they're held together by special bonds, and they're cross-linked and all that jazz. But that's all kind of lecture stuff, and y'all probably got that iterated to y'all quite a few times. But the important thing to know is that gram-positive cells are going to be purple, and gram-negative cells are going to be reddish pink colored. And you can see gram-positive right here, gram-negative right here. So the important thing is that a gram-positive cell has this very thick layer of peptidoglycan, while this gram-negative cell has a thinner layer of peptidoglycan. Gram-negative cells also have a membrane covering them, in which are composed of these lipopolysaccharides, and these lipopolysaccharides are very important in this whole process of gram staining and differentiating between gram-positive and gram-negative cells. So let's take a look at what these stains actually are. So the vocab that we're going to be dealing with are primary stain, mordant, decolorizing agent, and counterstain. So your primary stain is that initial stain that you put on everything. So your overall picture, you put a stain on everything. This is your primary stain. Your mordant, now that's not as conceptual. And so, that's not as intuitive. So I don't know what mordant does. And so this is an important skill when I was studying for the test, is that you look it up. So if you type in mordant into Google, you'll get a definition that says a substance, typically an inorganic oxide, that combines with a dye or stain and thereby fixes it in a material. So that's just what it does. Is that it fixes the primary stain into the cells. And that's an important skill to do when you're studying, is to look up stuff that you don't know, ask questions, email me, do something, but don't just see stuff and then say, I don't know what that is, and then continue moving on. So that's what a mordant does. And so the next thing is your decolorizing agent. Your decolorizing agent allows you to remove the stain from one type of the cells. And then what you do is, now that one of the cells don't have a specific type of stain, then you counterstain it, and so you stain those cells that you decolorized. And so if we look at each of the different types of chemicals that we're going to be using. You're going to be using crystal violet, iodine, ethanol, and safranin. So the way to remember this order is PIES, P-I-E-S. So it's crystal violet, which is purple. It's a purple dye. Iodine is the I, ethanol is the E, and safranin is the S. Okay, and so your primary stain is going to be purple, your crystal violet. Your mordant is going to be iodine. Your decolorizing agent is going to be ethanol, and your counterstain is going to be safranin. And we're going to look at how each one works in this whole system. So if we go back here, what happens is crystal violet's going to enter in both types of cells. So crystal violet's going to go in here, and crystal violet's going to go in here. This is gram positive, this is gram negative. So what happens is after you add the mordant's going to go in here and then form a complex with the crystal violet that's already present. So you come in here and it forms a complex here as well. What happens here though is that the crystal violet iodine complex is too big to get past this thick layer of peptidoglycan. And so it can't escape this like matrix or this like the cell wall. And so it's going to remain in the cell. And so that's why these things are purple, and that the purple crystal violet stain didn't exit the cell. While here, what happens is this complex so in gram negative as well, the crystal violet will go in and the iodine will go in and form the complex. But what happens is because of the thin layer of peptidoglycan, it can actually exit the cell, but it will get kind of caught in this lipopolysaccharide matrix. And so that's what ethanol does. Ethanol comes in and disrupts this lipopolysaccharide matrix and allows the iodine crystal violet complex to escape and run from the gram negative bacteria. So what happens is you go back to your natural state of bacteria, and that, if you remember, bacteria are mostly water, 90%, and so it's going to be clear again. And so what we do is we flood the bacteria again with the counterstain safranin. Safranin will come into here and here as well. But the thing about safranin is that it's a lighter color than this crystal violet color, and so even though it's here, you don't see it because the purple color is too dominant to see any safranin color. While here, these cells are clear, there's no purple dominance here, and so the safranin color is actually kind of expressed in the gram-negative bacteria. Oftentimes, if you look at this closely, you'll see little specks of purple in here, and that's because sometimes the ethanol didn't get to the lipopolysaccharide matrix, or maybe in these specific bacteria you have a little bit thicker layers of peptidoglycan, and so it didn't really come out as much, or numerous other things. But the important thing is that the general gist of these bacteria are crystal violet, and are safranin colored, and so these are gram-negative bacteria. And so that's the theory behind what happens, or how these stains work. What's going to probably happen is that they're going to say, what happens if you don't have the primary stain? Like, what will happen to your cultures if you forget to add the primary stain, or if you forget to add the mordant, or the decolorizing agent, or counterstain? And so you'll have to go through and conceptualize, like, what happens. So if you forget to add the primary stain, obviously they're all going to be safranin colored, because none of them have that purple color for the mordant to complex with. If you don't add the mordant, all of the dye is going to get washed out, because that primary stain, it was small enough to get through that thick layer of peptidoglycan in the gram-positive bacteria, and so it's gonna, if you don't add that mordant, it's also going to be small enough to get back out of that crystal violet, of that peptidoglycan matrix. And so again, all of them are going to be safranin colored. If you don't have a decolorizing agent, you don't disrupt that that lipopolysaccharide matrix, and so the cells are going to be primarily purple, right? Because that complex wasn't disrupted, and they weren't washed away. And if you don't add the safranin, obviously all of them are going to be purple, because the gram-negative ones are going to be clear and you can't see them, and then the gram-positive ones are going to be purple because this whole process was just remain the same. So again, make sure you know what happens if you don't add one of them, or if you don't add two of the steps, or just go through each one of them and make sure you know what each one does and what happened if you didn't have them, because that's a big part of that test. So the big points here is know how to gram stain, know the order of the gram stain, in that it's pies, purple, iodine, ethanol, safranin, and then also know, also look things up if you don't know what, if you don't understand them, and then know how to do stuff like this. What happens if you don't have that? The last thing is go through your. You need to make sure to read this experiment pretty in depth, because I didn't go over how long you let each dye sit, and then also like some other factors. And so make sure you read your lab manual. It's page 47 through 49, and going through this with a lot of detail, because it's going to show up in a lot of spots in your practical and in your written portion.